Genetic diagrams, often referred to as Punnett squares, are essential tools in understanding the inheritance of traits, particularly those controlled by single genes. To grasp the significance of these diagrams, one must first understand the basic principles of genetics, which revolve around the concept of alleles. Alleles are different versions of a gene that can produce variations in a trait. For instance, consider a simple trait like flower color in pea plants, which can be determined by a single gene with two alleles, one for purple flowers and another for white flowers. In this case, the purple allele is dominant, while the white allele is recessive. This means that if a plant has at least one purple allele, it will display purple flowers. Only when a plant inherits two copies of the white allele will it exhibit white flowers. This dominance and recessiveness are fundamental concepts in genetics, helping to explain why some traits appear more frequently than others in a population. When we construct a genetic diagram, we start by identifying the alleles of the parent organisms. For example, if we cross a homozygous purple flower plant, which has two purple alleles, PP, with a homozygous white flower plant, which has two white alleles, PP, we can predict the possible genotypes of their offspring. In this scenario, all offspring will inherit one allele from each parent, resulting in the genotype PP. Since P is dominant, all offspring will exhibit purple flowers. This illustrates how a single gene can determine a visible characteristic and how dominant and recessive alleles interact. Now, if we consider a cross between two heterozygous purple flower plants, both with the genotype PP, we can use a Punnett square to visualize the possible combinations of alleles for their offspring. We set up the square by placing one parent's alleles across the top and the other parent's alleles along the side. This results in 4 possible combinations for the offspring: PP, 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 and PP. By analyzing this diagram, we can see that there is a 25% chance of producing a homozygous dominant offspring, PP, a 50% chance of producing a heterozygous offspring, PP, and a 25% chance of producing a homozygous recessive offspring, PP. This method of visual representation not only simplifies the process of predicting genetic outcomes, but also highlights the ratios of different genotypes and phenotypes that can arise from specific crosses. The beauty of genetic diagrams lies in their ability to encapsulate complex genetic principles in a straightforward manner, allowing students and researchers alike to grasp the underlying patterns of inheritance. Genetic diagrams also emphasize the importance of understanding the concept of independent assortment. According to Mendel's laws, genes for different traits can segregate independently during the formation of gametes. This means that the inheritance of one trait does not influence the inheritance of another. For example, if we were to introduce a second trait, such as seed shape, which can be round or wrinkled, we would create a dihybrid cross. In this case, we would consider the alleles for both traits, leading to a more complex Punnett square that reveals the potential combinations of traits in the offspring. In conclusion, genetic diagrams serve as a powerful educational tool, illustrating how characteristics controlled by single genes can be inherited. They provide a clear framework for understanding the interplay between dominant and recessive alleles, enabling predictions about the traits of offspring based on parental genotypes. By employing these diagrams, we can demystify the intricate processes of inheritance, offering insights into the biological mechanisms that shape the diversity of life. Through this understanding, we not only appreciate the elegance of genetic principles, but also recognize their profound implications for fields such as medicine, agriculture, and evolutionary biology.